Sunya.
Today we are going to talk about glycolysis, Krebs cycle, and electron transport system.
Glycolysis is the first stage of cellular respiration. In other words, glycolysis is the splitting of sugar, a process of releasing energy within sugar. There are two phases in glycolysis, which is the preparative phase and ATP generating phase. In preparative phase, a 6-carbon sugar will be reduced into 3-carbon sugar. While in ATP generating phase, ATP and NADH is produced.
In the phase 1 of glycolysis, which is preparative, then 6-carbon sugar is reduced into 3-carbon sugar. First, glucose is converted into glucose 6-phosphate with the presence of ATP and hexokinase. Then, glucose 6-phosphate is converted into glucose 6-phosphate with the presence of phosphoglucoisomerase. The fructose 6-phosphate is later converted into fructose 1,6-phosphate in the presence of phosphoglucokinase and ATP. Fructose 1,6-phosphate can be converted into dihydrosyl acetate phosphate and glycolysis.
At phase 2 of glycolysis, which is the ATP generating phase, glyceraldehyde 3-phosphate from the phase 1 glycolysis is converted into 1,3-bisphosphoglycerate by using enzyme glyceraldehyde 3-phosphate dehydrogenase. At this stage, NAD+ is reduced into NADH. 1,3-bisphosphoglycerate is then converted into 3-phosphoglycerate by phosphoglycerate kinase. 2 ATP is produced in this stage. Then 3-phosphoglycerate is converted into 2-phosphoglycerate by phosphoglycerate mutase. By using enolase, 2-phosphoglycerate is further converted into phosphoenolpyruvate, which is also known as PEP. At this stage, pyruvate is produced in the presence of pyruvate kinase.
Krebs cycle is also known as the tricarboxylic acid cycle, and it is a biochemical pathway that is used to generate energy to the oxidation of acetyl Coenzyme A. It is also used for the synthesis of NADH and for the production of amino acid. Krebs cycle takes place in mitochondria of eukaryote and cytosol of prokaryote. The first step involved is actually a preparatory step which begins with pyruvate. Pyruvate is derived through the glycolysis of glucose, which is a 6-carbon compound, and it is split into 2 molecules of pyruvate, which is a 3-carbon compound. The next step includes the oxidation of pyruvate into acetyl Coenzyme A by the enzyme pyruvate dehydrogenase complex. And in this reaction, a molecule of carbon dioxide and a molecule of NADH is generated. The acetyl Coenzyme A is 2-carbon compound. In the next step, the acetyl Coenzyme A combines with oxaloacetate, which is a 4-carbon compound to form citrate, and hence the resulting molecule of a 6-carbon compound. This reaction is catalyzed by the enzyme citrate synthase. Then, citrate isomerized into isocitrate by the enzyme aconitase. Isocitrate is oxidized into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase, and in this reaction, a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is generated. Hence, the alpha-ketoglutarate is a 5-carbon compound because 1 carbon is lost into carbon dioxide. Next, alpha-ketoglutarate is converted into succinyl coenzyme N by the enzyme alpha-ketoglutarate dehydrogenase, and in this reaction also, a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is released. Hence, succinyl coenzyme A is 4-carbon compound. Next, Succinyl coenzyme A is converted into succinate by enzyme succinylcholine. In the reaction, a molecule of GTP is generated. Succinate is converted into fumarate by succinate dehydrogenase. In this reaction, a molecule of QS2 is generated, which is used for the production of FADH2. Fumarate is then converted into malate by fumarase. And lastly, malate is converted into oxaloacetate by malate dehydrogenase, and there is a molecule of NAD reduced to NADH. Result of the Krebs cycle are 3 molecules of NADH, 1 molecule of FADH2, 1 GTP, and 2 molecules of carbon dioxide. Since the glucose is split into 2 pyruvate compounds, for each glucose there will be 6 NADH, 2 FADH2, and 2 GTP and 4 molecules of carbon dioxide.
Electron transport system (ETS) is the last component of aerobic respiration. It is a series of electron transporter embedded in the inner mitochondrial membrane that shuttles electron from NADH and FSDH2 to molecular oxygen. In the process, protons are pumped from the mitochondrial matrix to the intermembrane space, and oxygen is reduced to form water. Electron transport system (ETS) is the last component of aerobic respiration. It is a series of electron transporter embedded in the inner mitochondrial membrane that shuttles electron from NADH2 and FADH2 to molecular oxygen. In the process, protons are pumped from mitochondrial matrix to the intermembrane space, and oxygen is reduced to form water. To start, 2 electrons are carried to the first complex aboard NADH. This complex, labeled as 1, is composed of fluorine mononucleotide FMN and an iron sulfur FES containing protein. FMN, which is derived from vitamin B2, also called riboflavin, is one of several prosthetic groups or cofactor in the electron transport chain. A prosthetic group is a non-protein molecule required for the activity of a protein. Prosthetic groups are organic and or inorganic, non-peptide molecule bound to a protein that facilitate its function. Prosthetic groups include coenzyme, which are prosthetic group of enzyme. This enzyme in complex 1 is NADH dehydrogenase and is a very large protein containing 45 amino acid chain. Complex 1 can pump 4 hydrogen ions across the membrane from the matrix into the intermembrane space, and it is in this way the hydrogen ion gradient is established and maintained between 2 compartments separated by the inner mitochondrial membrane. Complex 2 directly receives FADH2, which does not pass through complex 1. The compound connecting the first and second complex to the third is ubiquinone, labeled as Q. The Q molecule is lipid-soluble and freely moves through the hydrophobic core of the membrane. Once it is reduced, ubiquinone delivers its electron to the next complex in the electron transport system. Q receives the electron derived from NADH from complex 1 and the electron derived from FADH2 from complex 2, including succinate dehydrogenase. This enzyme and FADH2 form a small complex that delivers electron directly to the electron transport system by passing the first complex. Since this electron bypass and thus does not energize the proton pump in the first complex, fewer ATP molecules are made from the FADH2 electron. The number of ATP molecules ultimately obtained is directly proportional to the number of proton pumps across the inner mitochondrial membrane. The third complex is composed of cytochrome B, another ferrum sulfur protein, and cytochrome C protein. This complex is also called cytochrome oxidoreductase. Cytochrome protein has a prosthetic group of heme. The heme molecule is similar to the heme in hemoglobin, but it carries electrons, not oxygen. As a result, this iron at its core is reduced and oxidized as it passes the electrons, fluctuating between different oxidation states. Fe2+, and Fe3+. The heme group molecule in the cytochrome have slightly different characteristics due to the effect of the different protein binding them, giving slightly different characteristics to each complex. Complex 3 pump proton through the membrane and pass its electron to cytochrome C for transport to the fourth complex of protein and enzyme. At this time, cytochrome C is the acceptor of electron from Q. However, Q carries pairs of electrons. Cytochrome C can only accept one electron at a time. The fourth complex is composed of cytochrome protein C, A, and A3. This complex contains two heme groups, one in each of the two cytochrome A and A3, and 3 copper ion, a pair of copper A and 1 copper B in cytochrome A3. The cytochrome hold an oxygen molecule very tightly between the iron and copper ions until the oxygen is completely reduced. The reduced oxygen then picks up 2 hydrogen ions from the surrounding medium to make water. The removal of the hydrogen ion from the system contributes to the ion gradient used in the process of chemiosmosis. In chemiosmosis, the free energy from the series of redox reactions just described is used to pump hydrogen ion across the membrane. The uneven distribution of hydrogen ion across the membrane establishes both concentration and electrical gradient, owing to the hydrogen ion positive charge and their aggregation on one side of the membrane. If the membrane were open to diffusion by the hydrogen ion, the ion would tend to diffuse back across into the matrix, driven by their electrochemical gradient. Many ions cannot diffuse through the non-polar region of phospholipid membrane without the aid of ion channel. Similarly, hydrogen ion in a matrix space can only pass through the inner mitochondrial membrane through an integral membrane protein called ATP synthetase. This complex protein acts as a tiny generator, turned by the force of the hydrogen ion diffusing through it, down the electrochemical gradient. The turning of parts of this molecular machine facilitates the addition of a phosphate to ADP, forming ATP, using the potential energy of the hydrogen ion gradient. All in all, the electron transport chain is the portion of aerobic respiration that uses free oxygen as the final electron acceptor of the electron removed from the intermediate compound in glucose catabolism. The electron transport chain is composed of 4 large multi-protein complexes embedded in the inner mitochondrial membrane and 2 small diffusible electron carriers shutting electrons between them. The electrons are passed through a series of redox reactions with a small amount of free energy used at 3 points to transport hydrogen ion across a membrane. This process contributes to a gradient used in chemiosmosis. The electron passing through the electron transport system gradually lost energy. High energy electron donate to the chain by either NADH or FADH to complete the chain. As low energy electron reduce oxygen molecule and form water. The level of free energy of the electron drops from about 60 kmol in NADH and 45 kcal per mole in FADH2 to about 0 kmol in water. The end product of the electron transport chain are water and ATP. A number of intermediate compounds of the citric acid cycle can be divided into anabolism of other biochemical molecules such as non-essential amino acid, sugar, and lipid. These same molecules can serve an energy source of glucose spike.